class, I have described about the molecular structure of DNA molecule, and we have looked about how DNA molecule is created or how DNA molecule is formed. But as I have described in the last class, DNA as well as RNA both are genetic materials in all living beings. However, RNA is genetic material in certain bacteria. Very fewer bacteria or very fewer viruses are found where RNA acts as genetic material. But DNA is genetic material universally, starting from bacteria up to human being. So I think you are following the students in my last class, and I have described about the structure of DNA molecule. So today, in today's class, I will deal about how DNA molecule is created, and thereafter I will look into how RNA molecule also created, because it is also genetic material for certain organisms. So as I have described in my last class, the DNA is a polynucleotide chain. DNA is a polynucleotide chain. Polynucleotide chain of a large number of deoxyribonucleotides. The number of deoxyribonucleotides present in a DNA molecule depends on the structure and variation of the organism. The greater the complexity of an organism, note, bhabilaba bhalke, hunilaba, or parbukot paile eto note kori laba. Organism etar complexity jimane bahi jabo, himane DNA complexity u bahi jay. The length of the DNA molecule and hence complexity of the DNA molecule is greater. The organism which is better complex, more complex. So this relation keep into your mind. But overall remember is that DNA is a polynucleotide chain. Wherever it occurs, whether it is in virus DNA form, DNA virus to power, human being to power, I mean mango plant to power, grass plant to power. Kintu zote na power DNA molecule to hoday into polynucleotide chain. Car polynucleotide about deoxyribonucleotides polynucleotide chain. Also, I told in my last class, collect class of my education to Malkokswa, you just looked at in my last class. Also, I have said that deoxyribonucleotides is ribonucleotide of DNA. That means it's made by 3 different kind of molecules. Can you recall back? First molecule is one kind of nitrogenous base. Second kind of molecule is the deoxyribose sugar, and third kind of molecule is the phosphate group or phosphoric acid. This is acidic component. Yet কেতিয়াবা তোমালোকে নিটৰ লেভেলত বা এইমছৰ লেভেলত বা ডিপিএমটি লেভেলত বা বিএইচইউ লেভেলত বা এফএমচি লেভেলত বিভিন্ন ধৰণৰ কুৱেশ্যন পাব পাৰা। এইখিনি ইম্পৰ্টেণ্ট আছে। Deoxyribonucleotide are 4 different types on the basis of the base type, nitrogenous base type. মই কালি কৈছো তোমালোকক nitrogenous bases are, in my last class I have spoken that nitrogenous bases are total 5 different types belonging to 2 classes. One is pasta mi pam type, DNA site type, RNA site type. DNA thokaita asile adenine, guanine, cytosine and thymine. RNA thokaita asile adenine, guanine, cytosine kintu uracil thymine ami RNA na pao. Tenedore thymine DNA pao, uracil RNA pao, uracil DNA na pao. Mone rakhiba. Thymine is a pyrimidine base, so also cytosine. Cytosine and thymine is a pyrimidine base, and adenine guanine is a purine base. But যেহেতু adenine guanine purine base geta DNA and RNA দুবিধতে থাকে, দুবিধ নিউক্লিক এসিডে থাকে, গতিকে purine base সাপেক্ষে DNA আৰু RNA মাজত কোনো ডিফাৰেন্স নাই। বুজি পালা? DNA আৰু RNA এৰ মাজত purine base সাপেক্ষে, with respect to the purine base type, there is no difference found between DNA and RNA. But a small difference found in respect of the pyrimidine base type present in DNA and RNA. That is, that overall pyrimidine bases are 3 types. Pyrimidine base 3 type found. Ekita hol cytosine, thymine, uracil. Kintu here cytosine to RNA DNA duta thake. Kintu thymine to DNA thake, uracil to RNA thake. Uracil DNA na thake aru thymine RNA na thake. Eto mora rakhiba. Gotike overall tomalke 4 base type DNA pala. Ki pala ge overall? Ekita hobo A adenine, G guanine, cytosine and thymine is all DNA bases present in DNA. Adenine, guanine, cytosine, uracil is all bases present in RNA. Adenine, guanine, cytosine, thymine DNA base, adenine, guanine, cytosine, uracil all RNA base. It is here to base type site as a kote Accordingly, there will be 4 different types of nucleotides present in DNA and 4 different types of nucleotides present in RNA. Cybit nucleotide DNA, cybit nucleotide RNA কি হবৰ বেস কৰি এই নাইট্ৰোজেনাছ বেস কেটাৰ টাইপৰ হবৰ বেস কৰি আৰু বাকি কিটা কম্পোনেন্ট ডিফাৰেন্স নাই মানে ডিঅক্সিৰাইবোজ সুগাৰ DNA পায় ফসফেট গ্ৰুপ DNA পাম কিন্তু রাইবোজ সুগাৰ আৰু ফসফেট গ্ৰুপ RNA পাম গতিকে adenine plus ribose sugar plus phosphate is one kind of nucleotide we call AMP. Guanine plus ribose sugar plus phosphate group another kind of nucleotide we call GMP. Cytosine plus ribose sugar plus phosphate group another kind of nucleotide. Uracil plus ribose sugar plus phosphate group another kind of nucleotide. Similarly, yet adenine plus deoxyribose sugar, guanine plus deoxyribose sugar, cytosine deoxyribose, thymine deoxyribose, along with phosphate group, and form DAMP, DGMP, DCMP, DDMP. গতিকে DNA এইখিনি মই কালি কৈছোঁ তোমালোকক DNA থকা 4টা নিউক্লিঅ'টাইড নাম ৰাখিবা কিন্তু লগতে এটা কথা মই কালি ক'বলৈ পাহৰিছোঁ এইটো হ'ল that nucleotides are trimers made by 3 kind of molecules কিন্তু নিউক্লিঅ'টাইড ফৰ্ম হ'বলৈ যাওঁতে যেতিয়া নিউক্লিঅ'টাইড ফৰ্ম হ'বলৈ যায় তেতিয়া কিন্তু গ্ৰেজুৱেলি যায় প্ৰচেছটো প্ৰথমতে বেছটোৰ লগত সুগাৰটো লগ লাগে তাৰ পিছতে ফসফেটটো লাগে তাৰমানে বেছ আৰু সুগাৰ যেতিয়া লগ লাগে তেতিয়া আমি ডাইমাৰ এটা পাম গতিকে ডাইমাৰ প্ৰত্যেকৰ কাৰণে বেলেগ বেলেগ হ'ব এতিয়া adenine plus deoxyribose sugar is a dimer, guanine plus deoxyribose sugar is a dimer, cytosine plus deoxyribose sugar is a dimer, thymine plus deoxyribose sugar another dimer. গতিকে এই ডাইমাৰ কিটা কিন্তু আমি নিউক্লিঅ'টাইড নকওঁ নিউক্লিঅ'টাইডছ বিলাক ট্ৰাইমাৰ হয় ফসফেটটো থাকিলে নিউক্লিঅ'টাইড কম গতিকে adenine plus deoxyribose sugar is a dimer এই ডাইমাৰ বিলাকক য'তে নহওক লাগে RNA বা DNA এই ডাইমাৰ বিলাকক কিন্তু নিউক্লিঅ'চাইড বুলিহে কোৱা হয় গতিকে I think you are following students I think you are following about the difference between nucleoside and nucleotides গতিকে তোমালোকে দুটা টাৰ্ম পাবা এটা হ'ল nucleoside one concept and the other concept is nucleotide and nucleotide the nucleosides বিলাক হ'ল dimer আৰু nucleotide বিলাক হ'ল trimer বিলাক these dimers are made by a nitrogenous base plus a sugar okay তেনেকে কিন্তু ট্ৰাইমাৰ হ'বলৈ হ'লে বেছ থাকিব সুগাৰ থাকিব প্লাছ ফসফেট গ্ৰুপ ডিফাৰেন্স মাৰিব তাৰমানে ৰিলেশ্যনটো কি হ'লগে নিউক্লিঅ'টাইড ইজ ইকুৱেল টু নিউক্লিঅ'চাইড প্লাছ ফসফেট বা নিউক্লিঅ'চাইড ইকুৱেল টু নিউক্লিঅ'টাইড মাইনাছ এ ফসফেট গ্ৰুপ এই ৰিলেশ্যনবিলাক মনত ৰাখি থবা আৰু যেহেতু মই কালি ক্লাছত তোমালোকে ক'লোৱেই কোৱা হ'ল অলৰেডি the DNA কোন নিউক্লিঅ'টাইড থাকে আৰু কোন নিউক্লিঅ'চাইড পাবা তেতিয়ালে এইখিনি লিখিব পাৰিবা adenine plus deoxyribose sugar is deoxyadenosine this is one kind of nucleotide তেনেকে guanine plus deoxyribose sugar is deoxyguanosine deoxyguanosine another nucleotide cytosine plus deoxyribose sugar another nucleotide it will go deoxycytidine and then I get thymine plus deoxyribose sugar equal to deoxythymidine you have nucleosides of DNA these are nucleosides of DNA likewise nucleotides of RNA also you find কিন্তু ইয়াত কি হ'ব এবাৰ deoxyribose sugar নাপাবা ইয়াত থাকিব ribose sugar G plus R C plus R U plus R গতিকে adenine plus ribose sugar is adenosine guanine plus ribose sugar equal to guanosine cytosine plus ribose sugar equal to cytidine uracil plus ribose sugar equal to uridine okay এতিয়া মন কৰিবা ইয়াত আমি প্ৰেফিক্স ডিঅক্সি কিয় ইউজ নকৰো কাৰণ ইয়াত সুগাৰটো ডিঅক্সিৰাইবোজ সুগাৰ ন গতিকে adenosine is a nucleoside কিন্তু adenosine monophosphate is a nucleotide guanosine is a nucleoside but guanosine monophosphate is a nucleotide Cytidine is a nucleoside. Cytidine monophosphate is a nucleotide. Uridine is a nucleoside, but uridine monophosphate is a nucleotide, and so it is. Which basically, I think this is very important, or you must know about this. I think you have accepted this. So this is what exactly the relationship between nucleoside and nucleotide. From a schooling point of view, these are very, very important. So overall, DNA is formed of a large number of nucleotides forming chains. Think about it. You have followed, you have written about this. Now, DNA is made of two chains, not a single chain. While RNA is made of one chain, some exceptions maybe there are exceptions. Exception DNA. In most cases, DNA is double-stranded. In most cases, DNA double-stranded, except one organism. We find later more examples of organisms, but now we are not entering into complexity. Remember, initially, that DNA double-stranded প্ৰায় সকলো organism except one organism where DNA is single-stranded. The organism virus is 5*174. 5*174 polyphase. Okay, it is a virus. That infects E. coli. Bacteria. Don't need bacteriophage. Virus or bacteriophage. E. coli polyphase. Polyphase is a bacteriophage. That host to E. coli. Escherichia coli. E. coli attack the virus. The virus to 5*174 polyphase. It is the known organism, most popular organism, where single-stranded DNA is formed. Here DNA is formed, single-stranded DNA. Single-stranded DNA was it was single-stranded DNA. Kintu baki organism starting from bacteria onward, but bacteriophage namor virus bilakor pora ami jodi start koru. Gotike bacteriophage, bacteria, all other monorans, protista, fungi, plantae, animalia. Jihok etamolke example nidia. Everywhere that DNA is double-stranded. Kintu DNA double-stranded hoy. A to bad DNA double-stranded. Kintu hoy to DNA. I mean, occurrence of that DNA can occur where DNA occurs. Where DNA occurs. Occurrence of DNA occurs. DNA code for the DNA is found. DNA we will find DNA is the more prominent genetic material. Prominent genetic substance. Okay, yet my term use is genetic substance, but genetic material is a substance which passes from a molecule, a biomolecule, which passes from one generation to next generation, parental generation to the subsequent daughter generations, carrying the parental characters to the subsequent generations, means daughter generation. Parental generation or daughter generation carry molecule genetic substance. They are responsible for transmission of parental traits from one generation to next generation. Genetic substance. Any material that functions this, perform this, is called a genetic substance. And DNA is a good example of genetic substance, and almost universally it is the genetic material of every organism except some viruses, HIV, Ross sarcomavirus, bad di example pisot pam gebo. Kitaman virus bad di pray hokol bilak organism bacteriophage ko arambo kori human being lake ami DNA genetic substance pam hol mainly that it perform transmission of parental characters from the generation to the next generation. Eta generation or another generation genetic material transform transfer function genetic substance. In genetic substance, it is genetic substance because it performs some functions of a genetic molecule. A genetic molecule or genetic substance Key character what are the characters that is not progress, is not progress. So DNA is a genetic molecule. Later we will find the proofs. Mokshat malko describe korim. Kisapre to kisapalda ase. The DNA is genetic material. How it has been proved? DNA is genetic material. How can I prove kora hol? অলপ পিছত আমি আহি আছোঁ কিন্তু কেমিকেল ষ্ট্ৰাকচাৰ কৰি থাকোঁতে মই দুই এটা কথা আনুষংগিক ৰিফাৰেঞ্চেছ হিচাপে আমি ইউজ কৰিব লাগিব, ক'ব লাগিব। এইটো হ'ল চোৱা ডি এন এ জেনেটিক ছাবষ্টেঞ্চ বুলি কোৱা কেইবাটাও কাৰণ আছে, ইনডিৰেক্ট প্ৰুফো আছে, ডাইৰেক্ট প্ৰুফো আছে। ইনডিৰেক্ট প্ৰুফৰ ভিতৰত কেইটামান আমি এজিউম কৰিব পাৰিম, কেইটামান কেৰেক্টাৰ এজিউম কৰিব পাৰিম। হ'ল নম্বৰ ওৱান ইজ দেট ডি এন এ ইজ কেমিকেলি stable molecule, chemically stable molecule, chemically as well as structurally, chemically as well as structurally stable molecule. এই property কোন RNA না থাকে, কারণ আমি জেনেটিক মেটেরিয়াল বলে নিউক্লিক অ্যাসিড জেনেটিক মেটেরিয়াল আমি করতে পারছি পিছত নিউক্লিক অ্যাসিড দুবিধ আছে RNA আৰু DNA। তো RNA জেনেটিক মেটেরিয়াল কিয় নহয় নোহোৱাৰ কাৰণ DNA কেইটামান প্ৰপাৰ্টি থাকে, সেইকেইটা প্ৰপাৰ্টি RNA নাই, আৰু সেই প্ৰপাৰ্টিকেইটা DNA থলে তুমি DNA প্ৰেছাৰো জেনেটিক মেটেৰিয়েল হ'ব। তাৰে এটা প্ৰপাৰ্টি হ'ল DNA ইজ কেমিকেলি এজ ৱেল এজ ষ্ট্ৰাকচাৰেলি ষ্টেবল মলিকিউল। কিন্তু ইমান ষ্টেবলো নহয়, চোৱা DNA কেন প্ৰভাইড means of transformation so that it can induce variation according to demand of the nature. কিন্তু এইটো নহয় স্যার, it's not although chemically or structurally stable molecule, but still yet the DNA, yet even if it is stable molecule, yet the DNA molecule is capable of undergoing slow changes in nature according to the nature's demand. And this is also under property to the DNA molecule, but absent RNA molecule. RNA molecule to human stable now. RNA is comparatively less stable. RNA less stable होगा कारण तो हमें किसी प्रश्न को DNA ऊपर आ जाएगा। That demand को खुद को पढ़ेगा demand, किसी नहीं पढ़ेगा देगा। Why RNA cannot act as universal genetic material. RNA universal genetic material is pure non-conformal. RNA cannot act as universal genetic material like DNA. DNA is conformal minimum. RNA is a labile molecule. Chemically unstable molecule. Chemically unstable. Unstable. Why unstable? Chemical unstability over DNA nucleation, RNA nucleation. Why unstable? Why unstable? RNA unstable here. RNA unstable why around number one. The first number one of why RNA is chemically unstable. Number one, it is single stranded and exposed molecule. Single stranded and exposed molecule. Okay. Secondly, RNA has two ingredients. Which are ingredients here. Just two ingredients. That are whole. Number one, the uracil, and secondly ribosome. Okay. Uracil and ribosome both are active Over two and tender. Deoxyribose sugar respectively. deoxyriboside respectively. Active uracil and riboside are respectively active over the thymine of DNA, and deoxyriboside is over deoxyriboside of DNA. Now, uracil of RNA is active than thymine of DNA. Riboside of RNA is active than deoxyriboside of DNA. Because of our process will not show. Uracil is a pyrimidine base present in RNA. Thymine is a pyrimidine base present in DNA. DNA RNA has a difference in the display. Uracil, RNA, thymine, DNA. Thymine is more stable than uracil. A riboside of RNA is more active than deoxyriboside. Deoxyriboside is more stable than riboside. It is active, it is less active. It is full inactive plus inert molecule. It will be a uracil active molecule, thymine inactive, completely inactive molecule. It will get full inactive by inert molecule. DNA, thymine and deoxyriboside of RNA. DNA stable or DNA favorable genetic material for these are the causes of why RNA cannot act as genetic material for most organisms, but DNA can do. DNA stable. And the information these are very important things to you from example you can do. Exam of Kalora with the important information from that. Okay, more for more basic DNA genetic work around the monoma. Describe the causes why DNA is favorable genetic material. RNA or protein or lipid or carbohydrate cannot exist. আমি তার প্রশ্ন বিষয় দিয়েছি তোমালোকক গতিকে খালি মনত ৰাখিবা DNA genetic material হিচাপে কিয় কাম কৰে বাকি RNA বা প্ৰটিনে কিয় নোৱাৰে বা লিপিডে কাৰ্বহাইড্ৰেট কিয় নোৱাৰে তাৰ কজৰ ভিতৰত কজ নম্বৰ 1 হ'ল DNA is chemically and structurally more stable molecule. Stable হোৱাৰ কাৰণটো হ'ল কি thymine আৰু deoxyriboside. যেনেকৈ DNA যদিও stable molecule হয় তথাপিও yet still the DNA molecule is capable of undergoing slow synthesis in nature but nature's demand. Natural demand of nature provision has to cope with the nature organisms can modify nature. And this is very essential because if the organism unable to undergo change according to changes in the nature, then organism will not be able to survive. কারণ এনভাইরনমেন্ট যদি চেঞ্জ হয় আমি যদি নিজকে রেজিলিয়েন্ট কৰি নলওঁ আমি যদি প্লাষ্টিক হৈ থাকোঁ ইলাষ্টিচিটি হিচাপে কাম নকৰোঁ তো আমাৰ দেশ হয়তো দেখিম যে উইল নট চাব গতিকে তাৰমানে DNA ইলাষ্টিক মলিকুল বুলি ক'ব পাৰিছোঁ যদিও ষ্টেবল হয় কিন্তু ষ্টিল ইজ ইলাষ্টিক ডেট ইজ দি নেচাৰ্ছ ডিমাণ্ড দেন দ্য DNA মলিকুল কেন আণ্ডাৰগো চেঞ্জ আৰু সেই চেঞ্জটো কাৰণে দিছ চেঞ্জ is called mutation. It is called mutation. আমি পিছত ডিসকাস কৰিব পাৰিম যে পিছত কম mutation মানে গতিকে unexpected change, disoriented change, that orientation now, disoriented or unexpected change that can occur in DNA molecule according to the demand of the nature to help the organism to mutate or change. Required for the survey is called mutation. Okay, so mutation DNA hobo pare or mutation actually this mutation is the fountainhead of all sorts of variation present all sorts of variation found in organisms Okay. এইখিনি হ'ল ভেৰিয়েশ্যনছ মানে কি আৰু ভেৰিয়েশ্যন DNA যদিও ষ্টেবল মলিকুল ভেৰিয়েশ্যন দেখাব পাৰে কিন্তু একাৰ্ডিং ডিমাণ্ড কিন্তু ভেৰিয়েশ্যনটো সিমান ডাঙৰ ভেৰিয়েশ্যন নহয় ইটছ নট ছ মাছ বিগাৰ ভেৰিয়েশ্যন ইট ইজ স্মল এমাউণ্ট অফ ভেৰিয়েশ্যন স্মল কাইণ্ড অফ ভেৰিয়েশ্যন দ্যাট DNA এক্সিবিটছ মনে ৰাখিবা দিছ ইজ ইম্পৰ্টেণ্ট ইনফৰমেশ্যন আমি পইণ্ট নম্বৰ 1 টু এইখিনি পালোঁ আমি হাৰ্ড কেৰেক্টাৰ Hard property, but hard character why DNA can act as antimaterial. DNA is most probably antimaterial. How could we respond to that? So DNA is capable of DNA is capable of undergoing faithful replication or duplication and able to transfer copy to next generation organisms. Next generation. Copy बनाबो पारे এটা DNA molecule আছে তাৰ এটা পাৰ্ট এটা প্ৰপাৰ্টি থাকে সেই এটা DNA molecule ডাবল হৈ পেলাই দুটা সেই DNA molecule produce কৰিব পাৰে same DNA molecule can multiply faithfully and transfer those DNA molecule to offspring so that the offsprings are produced identical to the parents. তাৰমানে we are human being, মই human আৰু my DNA is faithfully duplicated into 2 DNA and sent to two that are organisms that are সেই cell যেটা produce হ'ল যিয়ে মোৰ বডি পৰা DNA ল'লে সেই ডেটাৰ চেল কেইটা মোৰ চেলত এৰি যাব তাৰমানে organisms can produce its own copy because DNA have this property. DNA property was it faithfully it can multiply and send them to the successive generations in order to maintain the species. Or this is why we are human beings continuously surviving and generation after generation we are human. Third generation human, next generation human, third generation superhuman. So you have to know DNA faithful replication of the DNA molecule that it can faithfully multiply, Okay? So if you want to know how property and DNA character part is DNA character to set more characters of DNA will find later on, and for which DNA can be treated as genetic material. So why DNA acts as genetic material? That costing amount of video biology, it's very essential, or you must, I think you might have know that. Is it clear? DNA occurrence of DNA. Next part is DNA occurrence. Occurrence means since DNA is genetic material and genetic material must be contained by every organism, therefore the particular entity can behave as a living being. I mean, an organism living being DNA the DNA is universally found in all organisms. Because DNA the DNA occurs in every organism because it is the genetic material universally present in all organisms. You get occurrence to what DNA occurs as a genetic substance. It occurs in both prokaryotes and eukaryotes. DNA as genetic substance occurs in both prokaryotes and eukaryotes. Prokaryotes and eukaryotes developed was the organism. Eukaryotes organism DNA or DNA the DNA is found therefore in both prokaryotes as well as in eukaryotes. In prokaryotes as well as in eukaryotes, DNA occurs. So that way, now let me draw the DNA that occurs in prokaryotes. The DNA that occurs in prokaryotes is called naked DNA. The DNA in prokaryotes called naked DNA. Naked DNA. Okay? In comparison to eukaryotes, the DNA in eukaryotes is bounded DNA, not naked DNA. The cause is that in case of prokaryotes, in nuclear envelope, since in prokaryotes, a true well-defined nucleus is absent. A well-defined nucleus is absent in prokaryotes. So it is sure that DNA in prokaryotes is naked DNA. In eukaryotic cell, however, it is bounded because it is covered within the boundary of nucleus. From eukaryotic cell eukaryotic cell nucleus but I am speaking. Suppose it is an eukaryotic cell. If I draw eukaryotic cell, it is a prokaryotic cell. Lump sum diagram I am drawing you. The lump sum diagram of the prokaryotic cell. It's a lump sum diagram of a eukaryotic cell. In case of eukaryotic cell, you will find a well-defined nucleus having a well-defined nuclear envelope. This is nuclear envelope. This is same nucleus. This is nucleus. Then upon nucleus move the prokaryotic cell on the port. So the prokaryotic cell of DNA to DNA. This is DNA. This is the individual DNA. In prokaryotic cell, the DNA is this. That nuclear envelope transmitted on a part. This is the DNA in prokaryotes. Prokaryotes, where there is no nuclear envelope. So it is naked DNA. It is naked. Nuclear envelope 9. Okay. In naked prokaryotic DNA to what nucleoid. It is called nucleoid or O-chromosome. Or xenophore or incipient nucleus. The DNA in prokaryotes is number 1 naked. Because nuclear envelope is absent, nuclear envelope 9. Kintu in naked DNA of prokaryotes is also called as nucleoid, also called as prochromosome, also called xenophore, also called incipient nucleus. So I did not know this must you note down. Okay? In eukaryotes, the DNA present inside well-defined nuclear envelope. It is called nucleus. Nucleus to cipher like a nuclear envelope. Nuclear envelope of DNA. DNA occurs inside the nucleus in the form of chromosomes. This is a pair of chromosomes. This is another pair of chromosomes. This is another pair of chromosomes. This is another pair of chromosomes. Okay? You look, the human cell, if I consider this is a human cell, and upon a pair of chromosome 23 pairs of chromosome, side of pair only 4 pairs of chromosome I am showing here. But ultimately, eukaryotic cell of DNA chromosome is DNA occurs here inside. Here inside this chromosome, DNA occurs. So here a chromosome at a pair elaborate it, modify, expand it, magnify it under electron microscopic view. Then a pair of chromosome will appear like this. Chromosome pair of this is one pair of chromosome I am bringing out from the nuclear environment. Nucleus of electron microscope observe. This is the whole chromosome at a pair. Here, DNA so you just observe that DNA will occur inside this as coil molecule like this. DNA molecule. Here also, this is the DNA molecule. Definitely, there is a big difference between prokaryotes and eukaryotes, cellular organization between prokaryotes and eukaryotes in terms of occurrence of DNA genetic material. So, genetic material DNA present in prokaryotes as well as eukaryotes node also. Due to the nature of occurrence, the manner in which the genetic material DNA occurs in prokaryotes quite different than that of eukaryotes. In eukaryotes, we find the genetic material DNA occurs under the boundary of nuclear envelope, confinement of nucleus, whereas such nuclear concept is absent in case of prokaryotes, and DNA in prokaryotes therefore called naked, কারণ nuclear envelope নাই, কিন্তু naked or aorta count. So there is another cause of why DNA is called naked in prokaryotes. Just DNA naked হওয়ার কারণটা আমি aorta কব পাৰো এটো হল যে prokaryotes of DNA histone protein is a very important information. This must be noted. Second cause of why. Let me diagram the muscle over here. Let me rub this diagram to explain of why the DNA of prokaryotes is also called naked in case of prokaryotes. Naked buli koa karon ditiyo karon to hobo case. Second cause of why the prokaryotic DNA is naked DNA called that second the DNA in prokaryotes is also called naked DNA because second cause because DNA have no histone association, histone protein association in case of prokaryotes. So here bounded hotar ami naked buli nokon. Kitu upori additionally DNA in eukaryotes is associated with remain associated with histone protein. Histone means basic protein. These are basic proteins. Histone proteins. And a association to actually histone protein and DNA is the association. Non-pair DNA is deoxyribonucleic acid. That is acid. In histone basic protein है basic. It is acid. It is basic. Acid and base. Acid base with a negative positive. But here electrostatic surge that the DNA possesses is opposite to that of the histone protein. It is acidic, it is basic for negative, it is positive for mutual attraction. Attraction to a thread produced. Those threads we call the filaments or chromosomes. Basic histone proteins can attract the acidic DNA. Produces the threads we call as chromosomes. A thread we call chromosome. A chromosomes is the concept to eukaryotes applicable, prokaryotes applicable now. Chromosome concept is applicable to eukaryotes only, but not applicable to prokaryotes. So this is the basic difference between how the DNA occurs in prokaryotes and eukaryotes. DNA prokaryotes can occur or can থাকে এটা কিন্তু তোমালোকৰ স্কুলিংৰ কাৰণে খুব ইম্পৰ্টেন্ট ইনফৰমেশন. Please note that down into your notes provided you are taking this. The layers are তোমালোকে নোট কৰি দিয়া কিন্তু মাকো কৈছো the DNA in prokaryotes is naked. What are দুটা আছে? There are two causes of why DNA in prokaryotes is called naked DNA. Number one is that there is no nuclear envelope to bound the DNA in prokaryotes. Secondly, there is no histone association with the DNA. Eukaryotes on the other hand, the DNA is remain bounded within a well-defined nuclear envelope, as well as there is DNA associated with histone protein, basic proteins, and chromosomes are found. So we have chromosome concept of set chromosome co-chromosome prokaryote DNA and chromosome pro-chromosome. Okay, I think you are following up to here. I have described about the DNA molecule, the structure of the DNA molecule, how it is formed, and occurrence of DNA. I was continuing. DNA prokaryotic cell eukaryotic cell every organism, whether it is prokaryote or eukaryote, protein organism DNA palm. But cell DNA Inside the cell where DNA you will find. Cell of DNA number 1 point. DNA inside cell in case of prokaryotes occur as cytoplasmic DNA. In prokaryotes, in case of prokaryotes, DNA occurs as cytoplasmic DNA. But in certain bacteria, in case of certain bacteria, DNA can occur as extranuclear DNA also, as extranuclear DNA also. This extranuclear DNA that occurs in certain prokaryotes is known by the term plasmid. In second year, this concept is very, very important for any competitive exams. Plasmid. The plasmids are extra chromosomal nuclear DNA present in certain bacteria, not in all bacteria. In certain bacteria, plasmids are formed. Okay, plasmids are discovered by William Hayes and Joshua Lederberg. It is again a very important terminology. Joshua Lederberg and William Hayes discovered plasmids in certain bacteria. Details plasmids are extra chromosomal nuclear DNA by extra chromosomal DNA present in some prokaryotes. Kisuman prokaryotes extra chromosomal DNA plasmid William Hayes and Joshua Lederberg. Plasmid and equation plasmid secondly, prokaryotes nuclear DNA extra DNA plasmid. If it's a DNA nucleus nucleus in eukaryotes similarly, DNA nucleus inside mitochondria, or in case of plant cells, mitochondria as well as plastid. Animal cell plastid plant cell plastid plant cell and animal cell divided mitochondria. Animal cell mitochondria DNA plant cell mitochondria and plastid DNA DNA present in the cytoplasmic mitochondria and plastids are called cytoplasmic DNA. It is nuclear DNA, chromosomal DNA and nuclear DNA. Mitochondrial DNA, plastidial DNA, these are extra nuclear DNA. The extra nuclear DNA, for example, mitochondrial or plastidial DNA. Extra nuclear DNA I mean, obviously, subcellular organism. Extra nuclear DNA are found like prokaryotes. Circular DNA is circular DNA. In certain eukaryotes, DNA may be contained in extranuclear or cytoplasmic organelles, cell organelles. These are mitochondria and plastids. Mitochondria and plastids. So these are called extranuclear DNA, extra chromosomal DNA, extranuclear DNA, equivalent to plasmids. However, you cannot speak. Plasmid is equivalent to common. Dear students, today I have described up to this extent regarding the occurrence of DNA. Okay. In the next class, I will deal how DNA is framed of their double chemical nature and more properties of the DNA molecule that will be required for the separate details. Okay, thank you students. Up to you.